Tech Hub, Cambridge Science Revision made easy.
Hello students, welcome to the second session of our topic 1 on photosynthesis. And as we had indicated earlier, that in this session we are going to look at the scientific inquiry skills. 1, we are going to test the collection of, to test the gas produced in the photosynthesis process, that is oxygen gas. How do we collect and test for this oxygen gas? And number 2, we are going to look at an investigation on how light intensity affects the rate or the speed of photosynthesis. So let's dive in there. 1, when you are carrying out an experiment to show that it is oxygen gas that is produced in the photosynthetic process, definitely we should be able to collect that gas. And oxygen gas is collected by over water method. So we are going to use a hydrilla plant. That is a plant that is adapted to photosynthesize under low light intensity. And we have this form of setup. We have an inverted funnel. This inverted funnel is supported by small pieces of woods here so that the gas can get in and out of the water. And we have, of course, a test tube to collect this gas. So immerse the plant in water as shown in the setup. Then ensure that the test tube is filled with water. Then cover it up on this funnel. Leave it exposed to sunlight for a few minutes. After 3, 4, 5 minutes, we observe the bubbles of the gas, that is the oxygen gas being produced from this hydrilla plant as it photosynthesizes. And these bubbles of the gas are collected in this test tube. We are going to carry out a chemical test to ascertain that the gas collected here is oxygen gas. And to test for oxygen gas, we use a glowing splint. Take a splint, a wooden splint, put it on a flame. It will catch up the flame, but extinguish off the flame, but the splint should remain red hot. Insert it in the test tube that has the oxygen gas. If the glowing splint relights, that demonstrates that indeed the gas is oxygen gas. That is a simple experiment that is used to show that the gas collected in the process of photosynthesis is oxygen gas. The second investigation here, students, is to look at how light intensity affects the rate or the speed of photosynthesis. On my setup here, number 1, we must be able to identify the variables here. We are talking of the independent variable, the one that we are going to change in this investigation. Then the dependent variable, the measurable variable. Then we have the control variables, the variables that we need to keep the same in this investigation so that it is a fair test. In our investigation here, I'm sharing the diagram or the setup of the apparatus. We have a ruler. This ruler is going to measure the distance between the light source or the source of the light and the water plant. And basically, this is what we are going to use to vary the light intensity. The distance between the water plant and the lamp determines the light intensity. You move the lamp closer to the plant. That shows we have, of course, the light source will be of high intensity because it's closer to the plant. You move the lamp away from the water plant, and thus we are reducing the light intensity. Then how do we measure the rate or the speed of photosynthesis? We have a stopwatch here. We are going to count the number of gas bubbles produced after every 5 minutes, basically to get the number of gas bubbles for every 5 minutes. That will give us the rate of photosynthesis. Those are the variables. So 1, we are talking of the independent variable, that is the light intensity, and here it is varied by varying the distance between the water plant and the light source. And the dependent variable, we are going to look at the rate or the speed of photosynthesis. That is measured by counting the number of gas bubbles produced after every 5 minutes in this investigation. And we know of the principles of scientific inquiry skills. The control variables must be kept the same. So if you are doing a set of experiments, several experiments here, we need to ensure that the species or the type of the plant is the same. The size of the plant should be kept the same. The temperature should be kept the same. And the amount of carbon dioxide, so that we achieve fair testing. Then these control variables should be kept the same. But for reliability of the experiment, you're going to do this experiment at least 3 times, collecting the results and get the average. Then that will ensure that we have a reliable test. That is the main concept we need to get here when investigating how light intensity affects this rate of photosynthesis.